first topic is heredity and hereditary changes. In that topic, we are going to deal with the further following points. The first, here we should know the genes. Genes are the component that is present in our nucleus, helpful for carrying the hereditary characters from generation to generation. Because we also have the hands, our parents also have the hands. So, that is nothing but a transfer of the characters. Presence of the hands in our parents as well as in us, that is due to the genes. And hence, genes is also act as a unit of heredity. Here, the genes specially, it is going to carry the hereditary characters from generation to generation. Heredity is nothing but the characters which are going to shift from generation to generation is referred as heredity. The main, because it is related to the genes, the study of the genes, it is referred as genetics. And hence the father or pioneer of the modern genetics is John Gregor Mendel. The next scientist, that is Hugo de Veris. Hugo de Veris in 1901 he put forth the theory, mutational theory. In the further topic, we are going to deal what is the meaning of mutation. The one of the important sentence that we should remember, that DNA is our genetic material. In all living organism, it is acts as genetic material. Please remember, DNA is our genetic material, not RNA. But in case of viruses, because there is an exception for the genetic material in case of viruses, because in viruses RNA acts as a genetic material. So now, what is the full form of DNA? DNA is nothing but deoxyribose nucleic acid. Deoxyribose nucleic acid. And RNA is nothing but ribose nucleic acid. Now, one of the important thing that is present known as rDNA. So what is the full form? Recombinant DNA. Because we know that DNA is a double-stranded structure having the presence of genes on it. Because DNA is made up of chromosomes. So in that, there we can able to change the genes. Suppose we have to add some additional character. In that case, we can change the DNA base pairing, and that technique is referred as recombinant DNA. And why we study the heredity? There is somewhat reason why we study the heredity. Because we can diagnose any kind of diseases. Because if you are suffering from any disease, we are going to diagnose to the doctor. After diagnosis, we can have a treatment for that disease. And the third important, that is the prevention of hereditary disorders. Hereditary disorders, the disorders which are passing from generation to generation. Suppose if your parent is suffering from some disease and you are also suffering from the same disease, and that disease is referred as hereditary disorders. And hence it can be prevented by studying the heredity. And we can also have ability to produce the hybrid varieties. Because in case of the hybrid production, we are going to change their genetic material, and hence we can improve the quality of the plant or animals. So, and hence it is the applications of heredity. And all these things are written on the blackboard. You should remember, because it may come for the MCQ or object type of the questions. Next topic of the chapter number 1, heredity and evolution, that is protein synthesis. For the formation of a protein, the 3 essential steps are required that we had seen in the introduction, that is the transcription, translation and translocation. So in that, first of all, we should know what is the central dogma. Because as I told you, protein synthesis is nothing but the formation. Anything that is going to form. What is going to form? Proteins are going to form. So central dogma is nothing but synthesis of protein by DNA through RNA. Means DNA is required for the protein synthesis, but RNA is also one of the important factor. So central dogma is nothing but synthesis of the protein by DNA through RNA is referred as central dogma. So now one of the first process that is required in the protein synthesis, that is transcription. Transcription is nothing but the formation of RNA. Formation of RNA through DNA is known as a transcription. Means with the help of DNA, we are going to prepare RNA, and that process is referred as transcription. For the transcription, RNA polymerase is an enzyme that is required. First of all, we should know what is an DNA. What is it made up of? It is made up of sugar, phosphate and nitrogen bases. No doubt, DNA as well as RNA both are made up of sugar molecules, phosphate and nitrogenous bases. So what is the difference? As it is here, it is a D. D refers for deoxyribose sugar. Deoxyribose sugar, and RNA where R refers for ribose sugar. And nitrogen bases, in both the cases it is present, but it is in the form of purines and pyrimidines, as we had studied in the 9th standard. Adenine is paired with the thymine. Here guanine pairs with the cytosine. That is A paired with the T with 2 hydrogen bonds. These bonds are nothing but the hydrogen bonds. Guanine paired with the cytosine with the help of triple hydrogen bonds. But in case of an RNA, the thymine molecule, that is the T, is replaced by U. U is nothing but uracil. That is A always pairs with uracil with the double hydrogen bond. Guanine always pairs with the cytosine with triple hydrogen bond. So now let's deal with a short description about transcription. First, where it occurs? It occurs in the nucleus, because a cell shows a presence of nucleus and the surrounding material is referred as a cytoplasm. So first process, transcription, occurs in the nucleus. But as DNA is a double-stranded structure, means it have the 2 threads, so but only a single strand is utilized. How many? Only single. Here it is the structure of DNA. You can able to see the 2 strands are present. It is the enlarged one. First strand and second strand. You can able to see only one strand is utilized for the formation of RNA. The sequence is complementary to DNA. Here the RNA molecule is going to form, and the lines that are going to represent the nitrogen bases or nucleotide, you can say nucleotide. The nucleotide or that sequence will be opposite one. Complementary is nothing but opposite one. Suppose here A is present, so here it will be T. Here it is a G, here it will be C. That is always A pairs with U, because there is a formation of RNA, and we see that in case of RNA, A is replaced by U. Suppose it is a T on a DNA, so here it will be A, that is adenine. So it is going to form the RNA molecule in the nucleus. The sequence is nothing but A always going to replace by U, or T on a DNA, on RNA it will be adenine. If G is on the DNA, C will be on the RNA. If C on the DNA, G will be on the DNA. That is nothing but sequencing, always opposite one. Purines always get paired with the pyrimidines. A, G are the purines, T, C are the pyrimidines. Then the formed messenger RNA. The form. Because now the RNA is formed, it should be removed from the nucleus and enters into the cytoplasm. And hence, after the formation of messenger RNA, it comes into the cytoplasm. So, after the formation, what it brings, as it referred as a mRNA. mRNA, that is a messenger RNA. So, hence, it will bring the message. It will bring the message for coding the amino acids. Why? What we are studying? Protein synthesis. and proteins are nothing but the long chains of amino acids. But that amino acid should be coded, and that message it is bring about by the messenger RNA, by the DNA. And hence it brings the message for coding amino acid in the form of triplet codon. Always it is in a group of 3 nucleotides. Suppose ATG, ATG, GCT, it may be anything. Any sequence can be there. And there are the 20 essential amino acids are present in our human body. Suppose there are the 20 amino acids. For coding a single amino acid, a single triplet codon is needed. That is nothing but 3 nucleotides are utilized for the formation of single amino acid. By that, if you are going to form the 20 different amino acids, you will require the 60 different nucleotides. Why? Because if a single requires 3, then 20 will be multiplied by 3, that the answer will be 60, and hence 60 nucleotides will be required for coding the 20 different amino acids, because the triplet codon are the unique one, like a lock and key manner. So it is all about the transcription.